Today we are starting a very important series on antithrombotic drugs. But before we talk about aspirin or warfarin, we need to understand the system these drugs are trying to hack. We need to understand hemostasis. So, what is it? Formally, hemostasis is a complex process that results in a spontaneous arrest of bleeding from a damaged vessel, followed by a restoration of its function. But in simpler words, when a blood vessel is damaged, your body has a built in survival mechanism to stop the leak and fix the pipe. That survival mechanism is what we call hemostasis. It is a delicate balance involving four main players: the endothelium, platelets, coagulation factors, and the fibrinolytic system. If this balance tips one way, you bleed. If it tips the other way, you get thrombosis. To make this easy to navigate, let's break hemostasis down into four distinct phases. First, we have vasoconstriction. When a vessel is damaged, its immediate reflex is to constrict. This spasm serves two purposes. It lowers blood flow to reduce the leak, but it also concentrates pro-clotting factors in that local area so they can work faster. Second, we have formation of the platelet plug. This is where the action really happens. When the vessel wall is broken, collagen fibers underneath are exposed. Normally, platelets float by smoothly, but they have specific sensors that detect this exposed collagen. The moment they touch it, they stick to it immediately. Once they stick, they get activated. They change shape and release chemical signals to call for help. When those signals go out, new platelets arrive and stick to the first platelets. They start clumping together to form a physical plug. This whole sequence, adhesion, activation, and aggregation, is what we call primary hemostasis. And because the final result is platelets aggregating together, the drugs that target this phase are called antiaggregants or antiplatelet drugs. Third, we have stabilization or secondary hemostasis. The formation of the platelet plug is fast, but the plug itself is soft and unstable. To fix this, the body activates the coagulation cascade. Thrombin converts fibrinogen into a hard fibrin net and wraps around the platelets and traps the red blood cells. This locks the clot in place. We call this secondary hemostasis. But clinically, this is what we refer to as coagulation. And finally, the phase 4 is fibrinolysis. Once the vessel is fully healed, we don't need the clot anymore. The body uses an enzyme called plasmin to dissolve the thrombus and restore normal blood flow. Why does this matter for pharmacology? Because our drugs target these specific phases, drugs that stop aggregation phase are antiplatelet drugs. Drugs that stop coagulation phase are anticoagulants. And drugs that trigger the final cleanup phase to dissolve clots are fibrinolytics. Now, let's talk about the term antiplatelet. In English, that is a standard term for these drugs. But if we want to be scientifically precise, in many European languages this is the standard. It makes much more sense to call them platelet aggregation inhibitors, or antiaggregants for short. Why? Because the term antiplatelet sounds like we are attacking or killing the cell, like an antibiotic kills the bacteria. But we aren't. We are just stopping them from holding hands. aggregating. So while I use antiplatelet in this video, remember that antiaggregant is the better description of the mechanism. Another common question I get, can we call them thrombocytes? The answer is yes. Thrombocyte is a perfect synonym used in many languages. However, if you want to have a deeper biological understanding, to truly grasp the science, you should know the difference. In birds and fish, the clotting cells have nucleus. They are true cells, so they are scientifically called thrombocytes. But in humans, our clotting units do not have a nucleus. They are fragments that broke off from a giant mother cell called megakaryocyte. So platelet, which means little plate, is biologically more accurate term. Here is the best analogy. Think of red blood cell like a house where the owner moved out. It ejected its nucleus. It's still a house, but a platelet is like a small shed built from a scrap wood or a different giant house. It was never a house on its own. But clinically, you can use either word. Finally, we must answer the golden clinical question. When do we use antiplatelet drugs and when do we use anticoagulants? It depends entirely on the type of clot. First, we have a white thrombus, which forms in arteries. In arteries, blood flows fast. When atherosclerotic plaque ruptures, platelets are the main heroes, because they are the only ones sticky enough to hold on against that high flow. The resulting clot is rich in platelets, so we treat this with antiaggregants. This is why we use them for heart attacks and strokes. Second, we have the red thrombus, which forms in veins. In veins, blood flows slowly. This gives coagulation factors plenty of time to pull together and build a massive fibrin net that traps millions of red blood cells. So we treat this with anticoagulants, like Warfarin or Heparin. This is why we use them for deep vein thrombosis. So remember, artery problem means platelets, so we use antiplatelets. Vein problem means fibrin, so we use anticoagulants. So we have established that to stop arterial clots, we need to stop platelets from sticking together. But how exactly do they stick? How does a floating platelet suddenly turn into a sticky, spiky ball that grabs onto its neighbors? And more importantly, which specific receptors on the platelet surface can we block with drugs? To answer that, we need to zoom in. In the next video, we are going to dive deep into physiology of platelet activation, mapping out the exact targets for aspirin, clopidogrel, and others. See you there!